everyone. If everyone can take their seats now, please. All right, we'll begin very soon. If everyone can take a seat, please. We'd like you to know that this is being streamed live on synbiowatch.org and kpfa.org. So good evening. Good evening. Thank you very much for coming. I'm Tina Stevens of the Alliance for Humane Biotechnology.
Can you turn up the mic?
Can't hear.
How does one turn up the mic? I'll just talk to you from down here. Is that okay? It's not okay for me, but it's okay. How about up here? No. But want to thank you very much for coming and offer particular thanks to the CS Fund, whose generous grant made this evening possible. Scientists working on recombinant DNA research in the 1970s triggered public attention when they called for safeguards to protect against potential biohazards. But when the public responded, sometimes with alarm, the scientific community sought retreat from citizen scrutiny and secured methods of self-regulation. Since then, powerful financial incentives emerging in the 1980s reinforced this self-regulatory system, a system lacking adequate transparency or public participation. But the watchdog groups that emerged in the wake of early controversies never stopped watching. Last year, when plans were announced for the expansion of the Berkeley National Lab, a number of these organizations, urging responsible science, not anti-science, took note. The expansion would result in the world's largest synthetic biology lab. Yet there was little in the lavish promotional campaign wooing cities that suggested as much. Known informally as extreme genetic engineering, synthetic biology is controversial. Last year, 58 organizations from 22 countries signed an open letter to the President's Bioethics Commission disapproving of its lax recommendations on synthetic biology. Earlier this month, Friends of the Earth published Principles for the Oversight of Synthetic Biology. Signed by over 111 environmental, consumer, religious, scientific, worker safety, and human rights groups from around the globe, this document calls for mandatory synthetic biology-specific regulations in order to protect public health and the environment and to ensure worker safety. Yet, public promotion of the lab's expansion did not address any of the underlying concerns that resulted in these extraordinary collaborations. The consortium of civil society groups convening tonight's forum seeks to balance hyped claims and unmask concealed risks. It seeks to give public information needed to make civic participation possible in order to sort through urgent questions. What communities are being affected, and how are they being affected? How can these communities affect choices that are being made? How can they affect outcomes? Governance, oversight, transparency, regulatory integrity. What constitutes meaningful participation? Tonight's meeting is dedicated to the late historian of science, Charles Weiner, who, After skilled chronicling of the social unrest triggered by emerging genetic technologies 4 decades ago, stressed throughout his distinguished career the necessity of answering these questions. It is dedicated also to the often invisible injured biotech workers who are sometimes sickened and even killed by risky experimental research. We are joined this evening by a family member of Dr. Malcolm Casadaban, who died from a modified strain of the plague after a lab exposure. His experience and that of his family, and the experience of other lab accident victims and their families, remind us that concern about the human costs associated with the technologies that we pursue is as much a matter of securing and maintaining best practices and just redress during their development as it is of anticipating their social effects. To help us begin our consideration, the director of the International Center for Technology Assessment, Andrew Kimbrell, will moderate tonight's presentations. Andrew?
It is not often that we have a public forum like this. The first in my experience where the topic is above the fold on the front page of the daily newspaper. This is pretty remarkable. And as we look at this technology, and as someone who spent many years in technology assessment, I think it's important for us to remember that not to approach this technology, which is either a new technology, as you'll hear, or an application of an older technology, with what I call technological amnesia, or technology amnesia. You know, this is not the first technology that has come to us over the last several generations. We have had the nuclear technology. We have had the chemical technologies, right? We have the combustion engine. And when we look back on that history, what becomes very, very clear is that when they first started the first X-rays, they did not know that we would be, for many decades, under the cloud of nuclear destruction through nuclear weapons. They did not predict that technology was going to go there. The first few chemicals, they had no idea that we look, as we do at the world today, with toxic waste dumps and literally millions of new molecules out there that were never thought of before. Certainly when we started the company, as a person who grew up in New York City, no one imagined what would happen to the combustion engine if you've ever been on the Long Island Expressway at 5 o'clock in the afternoon. No one could have predicted that, much less the impacts, global warming, etc., that those causes. So let's not approach this technology. So let's not approach this technology with sort of amnesia. This is not the first technology that's been around the barn. We know that we have never limited technology to its beneficial purposes. We failed at that as a society. And we've never been able to adequately predict the consequences of the technologies we've implemented. So this forum also gives us a very unique opportunity with this technology to try and get ahead of the curve, both here in this room as a society and both here in this country and globally, on this new technology or amplification of old technology. This is a very unique opportunity. And fortunately, we have just the right people to help us get into that conversation. We know that it's also extremely topical here. We know about the lab in Richmond, which will be a topic of conversation both among the speakers and I'm sure in the discussion. Emeryville, of course, is corporate headquarters for a couple of the major synthetic biology firms. And we also know that Berkeley's long-term plan for development. The discussions of that have been dominated by our Secretary of Energy, Stephen Chu, and many of those in the synthetic biology community. So this is extremely topical. This is ground zero. Very important discussion for this community. But ground zero for what? What is synthetic biology? I guarantee you that the vast majority, the vast majority of the people in these communities could not answer that question. They're going to be asked to decide whether this is the right future for their communities, but they will not know what synthetic biology even is. So it is very appropriate that our first panel today is going to answer that question. What is biotechnology? I'm trying to give us a feeling for what this technology is and what it isn't, and what the scope of it may or may not be. And it's equally appropriate that our first speaker is Jim Thomas. Jim is a research program manager for the ETC group. And I have had the pleasure of being on many fora with Jim. And I can say that Jim is unquestionably the world's leading person in making the public aware and educating the public about this technology. So you're starting, I think, with the leading educator and public awareness expert in this technology. Jim?
Thank you, Andy. Thank you. I'm going to load up the PowerPoint, so you'll hold with me for a second. And thank you very much for so many people coming out. As Andy says, sorry, how do I start this? As Andy said, I work for an organization called the Etc. Group. We're a very small organization. We track emerging technologies. And synthetic biology is an area we've been tracking, on behalf of civil society groups probably for about 8 years or so. And looking around this room, I'm reminded of another meeting on synthetic biology that happened in Berkeley that I watched via a streaming camera, I think back in 2006, where there was a town hall meeting on synthetic biology and trying to discuss how do you regulate and what are the issues raised by synthetic biology here in Berkeley. And what was noticeable was that meeting it had 1 or 2 people in the audience. It had a few people up on the panel who were almost entirely from the synthetic biology community. The critical mass wasn't there yet to discuss the issues around synthetic biology, so it's good to see that critical mass is now beginning to emerge. And I think they have a very hard task here to describe very quickly what synthetic biology is. The President's Bioethics Commission last year, the question came up, what is synthetic biology? And I think it was Drew Endy from Stanford, who some of you will know, said, listen, chill out, you're not going to get a definition on this one. And as many of you, there are a number of people here in the room who are in the synthetic biology community. I recognize people who know this very deeply, so hopefully we'll have a bit of a discussion on that. And following me is going to be Professor Ignacio Chapela, and I think he and I are going to give a bit of a yin-yang presentation. I'm going to suggest what synthetic biology is, the standard version. I'm going to tell you the Wikipedia entry. Professor Chapela, a practicing microbiologist at UC Berkeley, will talk a little bit more about what it really is. And so some of what I'm going to say, some of the language, some of the fantastical claims I'm going to make, please do have a little skepticism in the back of your mind. There's a lot of language and metaphors here that we may want to test how real they are. But I think one thing that's indisputable is what synthetic biology is. Actually, it's less than a science and technology. It's an industrial plan. It's an industrial strategy. It's a plan to use biology and the engineering of biology to make stuff biologically. And what's very clear is that there is an actual synthetic biology industry taking root specifically here in the Bay Area, but other places too. And that industry, I'd say there's somewhere around 50 to 100 maybe synthetic biology startups. So maybe as many as that. Many of them are still in stealth mode. And many of them are right here. Here's a map we put together of the Bay Area labs that are synthetic biology labs and the companies. It's a long list of companies here, over 20. And I'm sure there's many missing there. Some of you will know which ones are missing. A lot of these are companies that are attempting to see if they can use the engineering of biology, a set of extreme genetic engineering techniques in order to make biofuels, bioplastics, natural products in microbes and so forth. And behind them is a number of investors, and particularly some of the world's largest companies. So if you look at the top 10 energy corporations, here they are, 6 of those. 6 of the top 10 energy corporations at least have investments or joint ventures in synthetic biology companies. Here's the top 10 chemical companies. 6 of them have investments or joint ventures with synthetic biology companies. One of them, I'd say, DuPont, increasingly is a synthetic biology company. There's so much of its strategy looking forward is about using synthetic biology to make bio-based materials. And we could call this presentation the Joy of 6, because here's the top 10 agribusiness companies. 6 of those as well have significant investments in synthetic biology. And in fact, pharmaceuticals are the ones that break the mold. So the top 10 pharmaceutical companies, 7 of those have investments in synthetic biology. And what does that look like on the ground? So some of you will know Jay Giesling. He's the head of JBay, the Joint Bioenergy Institute, that's going to be moving to the Richmond Lab, and probably at the center of the Richmond Lab. Also one of the leaders of SynBio, the Synthetic Biology Engineering Research Center of the National Science Foundation, is meeting this week here in Berkeley. He's head of biosciences at Berkeley Lab. Jay probably represents what's happening a lot in synthetic biology. He's founder of 3 companies, Amyris Biotechnology, LS9, Lygos. They produce various biofuels and biomaterials. He's on the board of 2 other companies, Evolver, which takes a bunch of defense money in order to make things like vanilla, Genomatica. This was what Jay said this morning in the San Francisco Chronicle about the new lab. As the charge that the new Richmond Research Center will be dominated by corporate interests, he said it will remain completely independent of the energy industry. I'm finding this an astonishing claim. Here's if you go to JBay's website, where they have a whole section called For Industry. So this is their strategic partners, Total, Statoil, BP, the Industry Advisory Committee, LS9, DuPont. These are energy companies. This is who's going to be moving into Richmond. And JBay is very clear about this. They say, we work with companies because they can then leverage our facilities. JBay says, we will give them intellectual property, first dibs on intellectual property. We will give them the chance to get a significant return on investment. In fact, this is a cheap way for these companies to do research, in this case in biofuels, on the public tap. And Amyris too. I think we have to look at there is a conflict of interest here. Jay boasting again to the San Francisco Chronicle that his 1 million shares are worth about 17 million in Amyris. Well, here's who Amyris has partnered up with. Let's see, Shell, Total, these are energy companies. I think the point is that what we're seeing here is the beginning of a network of companies using this technology. That, interestingly, these are not just energy companies. You've got chemical companies, you've got cosmetics companies, you've got grain companies. This technology, supposedly synthetic biology, begins to cut right across all these different areas. So why all this corporate money flowing around or buzzing around this synthetic biology thing? Well, there's 3 promises or 3 claims that I think the synthetic biology community are making that they might be able to do, 3 possible magical tricks. The first is that they might be able to turn microbes, yeast and coli and even algae, into little chemical factories, and I'll touch on that in a second. The second is maybe they have the technology to turn sugar into oil. And the third one is maybe they can make nanorobots, broadly speaking. So microbes as chemical factories. You hear this a lot when you look at the literature from companies like Amyris. This is yeast, brewer's yeast. It's a magical little bug that's able to make wonderful things like beer and wine. But the notion is if we can reprogram it, if we can put in new genes in a particular order such that it, instead of making ethanol or beer or wine, why couldn't it make something more like gasoline or jet fuel or something more like plastic? And so you have this idea that these are tiny factories that eat sugar and spit out an industrial commodity. And then these are factories that reproduce, so you have a lot of them in a vat in something like a brewery. So that's the business plan for a lot of these synthetic biology companies, re-engineering these tiny little factories. In particular, it's interesting if you can do that, and you can spit out something like petroleum. Well, then you're doing a magical thing. You're turning sugar into oil. And in a time when petroleum is becoming harder to access, and petroleum is becoming more expensive to access, and politically more expensive to access, then if sugar can become a new petroleum field, this is extremely interesting to the likes of Total and Shell and BP. Because those sugars include cellulose, which is the woody sugars that you get in forests. And if you can access those sugars, then all of the black parts of this map, those are the forests, those are the biomass. 86% of living plants are in the tropics here. All of those become above ground oil fields that become open and available. So sugar becomes the new oil, as Bidexis, one of the synthetic biology companies, points out. And that's very much in the plan. There's Steve Koonin, who previously was at UC Berkeley leading synthetic biology work. Previously he was with BP. Now he's at the Department of Energy. And yes, we'll go for the green parts. So the green parts are where we want to go. It's the forests. There's even a suggestion maybe we can turn carbon from the air, carbon dioxide into oil using algae. There are real limitations on this, actually. But the idea is if we can engineer algae and put it in large ponds, maybe that will create an oil. Algae does produce oil, but there's synthetic biology potentially could make algae more efficient at producing oil and make it in a different way. And then this idea of tiny nanorobots. Drew Endy from the University of Stanford. Programming DNA is cool because you have actual living machines. That's what viruses are, something like that. Bacteria. And if you could reprogram them to start attacking different parts of the body, it's like programming a little machine, a molecular machine. We'll come back to this later. So what's the actual technology here? Well, it's this notion of these are machines, that these are factories. If they really were factories and machines, then you would have to have some instructions. And the instructions, of course, for living organisms is supposedly DNA. It's letters G, T, C, C, A, T. And then if you could rewrite those letters, which is what genetic engineering has tried to do. It's tried to cut out bits of DNA from one organism and put them in another in a kind of cut and paste fashion. But if you could just write it, you could write it like you were on a typewriter, then you could make your own instructions. So this is a machine called a DNA synthesizer. It's about this big. There are 4 bottles of 4 chemicals, G, T, C, and A, and roughly speaking, it prints out DNA, and you have synthetic DNA. And you can then say, I want a piece of DNA that goes G, T, C, 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 A, T, C, and there it is. It just makes it for you. So you don't have to go and find it in nature. You can just invent it. And then you can put it into an organism and program the organism. In fact, you don't even really need a machine because you can go online to someone like DNA 2.0 who are based down in Silicon Valley, and you can type in, I want GTTCCCA, and they'll send it to you in the post at about 30 cents per letter. And so that means you can just order DNA that you want, and you can use that for supposedly programming these things. And so within synthetic biology, the idea is that we can make this a standard process. We can have standard commands for that program, so-called bio-bricks. And you can then put those commands together like you make circuits. So of course, Silicon Valley was built on building circuits into computers, and a whole industry got built on the back of that we now see with Apple and IBM and Google and so forth. And the idea is here we're going to build biological circuits and build an industry on the back of that as well. That's the dream for synthetic biology. And pushing that dream is things like the International Genetically Engineered Machine Competition that happens every year. I think it happened at Stanford most recently, where you have hundreds of high school students and undergraduates coming together to build synthetic organisms, to build artificial organisms that flash different colors or smell of bananas and so on. And it doesn't have to be done by high school students. It can be done by people at home. This is a woman called Meredith Patterson here in the Bay Area. She calls herself a biopunk. Here she is in her kitchen producing a microbe that will glow green in yogurt. And there's a whole movement called DIY Bio. It's particularly strong here in the Bay Area. There are community labs doing synthetic biology. It doesn't need to be LBNL's lab down in Richmond. Bill Gates thinks it's a really great thing. And of course, you can go beyond printing out these little strands. So here's the full DNA sequence, all the letters for making the polio virus. 7,500 letters. You could theoretically put that into a sequencer, and you could print that out, or you could get all the bits and you could put them together. There's a guy in New York who's done this several times, Eckhard Wimmer. He's built the polio virus probably 7 or 8 or 9 times now, probably more than that. And it works. You put it into a mouse, and the mouse dies. Or you've possibly heard about this. This is work done by Craig Venter, where he built the entire genome, all the instructions, the DNA instructions for a bacteria, and booted it up, made it so that the bacteria reproduced. Took all the instructions for a goat bacteria, a goat pathogen. He called it JVC001. We call it Cynthia. So that's it, just a very quick run through. What is synthetic biology? It's a new industry that tries to reprogram living organisms, particularly microbes, but not just, there's also plants. The idea of turning them into living factories that will turn sugar into oil or into plastics and so forth. I'll leave it there. Thank you.
and that was great. Our next speaker, it's an honor actually to introduce our next speaker, Ignacio Chapela. It is never easy to speak truth to power. Ignacio knows that. Several years ago, he discovered the contamination of traditional maize in Mexico by genetically engineered varieties, wrote a scientific paper on it, and was attacked by corporations and attacked by people inside his own university here at Berkeley. He succeeded. He was able to defeat those who wanted to suppress his science, and since then has become a staunch, really the conscience in many ways of Berkeley, and as we have seen a continuing takeover, not just of Berkeley, but around the country, of universities by corporate interests. And he has been raising the red flag whenever that's happened here at Berkeley, which is unfortunately way too often. One of the major reasons we want Ignacio to speak next, though, is because as someone who's been looking at this science and technology for almost 30 years, it is my experience that Ignacio is at the cutting edge, really, of understanding what's really going on with genetics, what is hype, and what is truth. And he's going to share that with you right now.
Time is very limited. So, thank you very much. Please raise your hands when you don't hear when I start drifting away from the microscope and you cannot hear. And I'll try to speak louder and come back to it. It is a real privilege to be here in front of you. In more than one way, I'm really privileged to be among the very few faculty members on our campus who would even dare set foot in this meeting. I'm proud to see that there are a couple of them in the audience, and that's great. But one of the things that we shine for is our incredible silence as critical voices. And I wish I could be here to speak and detail about our technology. But I'm afraid that I do not even have the... I'm sorry. Let's hope that I have a couple of photographs that I would like to show you, even though time is very brief. Okay, it's not going to work. I can see that. Let's just stop that. That's good. Thank you. I wish I could be talking about our technology, but I don't have the heart to represent to you is that coming from the projector? You think? Okay, that's, very powerful people really love to see themselves projected in that. And there's something really eerie about seeing in my background Microsoft. That's why I'm happier just to have it. I understand. Yes. That would certainly not boost my ego. Our technology, are we talking about our technology? The reality is that we're not. The reality is that DNA, no matter how we synthesize it or not,
take it out.
whether we synthesize it or take it out from an organism and purify it, no matter where it comes from, it is a really boring molecule. You put it in a tube, you give it all the materials that it needs to do whatever you think it's going to do, and it does nothing. It is really the context of DNA that, the living context of DNA that people use to piggyback on it, to produce the things that they think they're going to produce, and to sell the things out there that they think they're going to sell. But most of what they're selling are ideas, are promises, are hopes of an application that really hasn't happened. We continue to hear that we're always 5 and 10 years away from the next great product and the next great breakthrough, and finally we're going to be able to continue to run our cars on biofuels and so on and so forth. But the reality is that the history of biotechnology is as old as the history of nuclear power. It all starts with Vannevar Bush and the idea that we can have the never-ending frontier of development, innovation, development, and technological application that would drive the world through economic development. And while the nuclear power that was envisaged back in the 30s actually delivered, and Hiroshima and Nagasaki were very early proofs that it works. And nuclear power is a very good proof that it works. At the same time, a parallel track to run the biotechnological dream continued to draw blank. And to this date, even though these companies do sell stuff, they sell gallons of fuel at $50 and $100 a gallon, and they have the Navy to buy it so they can continue their business programs and they can continue to raise venture capital and so on. Even though they sometimes can produce some things, they continue to run at a loss. If you ask the real, the place where the pudding is shown, which is really the markets, which is really where people want to put money into it or not, you will see that even though we have invested over 40, 50 years into this enterprise, more than 300, depending on who you ask, hundreds of billions of dollars have gone into it. And we're still yet to make a profit. And we're still yet to make a really predictable, controllable living organism that will follow design the way engineers would like it to follow. So I cannot really speak about a technology with a clean conscience. I can speak about a form of engineering, though. It's a very important kind of genetic engineering that happens and has happened and has been very successful, and that is the genetic engineering of your public institutions. Through a process called PPP, public-private partnerships. And this is a very appropriate place to speak about it because we at Berkeley many people have been very proud to be the pioneers of PPP. We started welcoming these private-public partnerships as strategic alliances, what they were called, in the 90s, as a dry run to what became in 2007, 2008, a really model for the world on how to do this. With PP, we obtained $500 million that really very quickly distributed throughout our campus and had a very effective outcome leading to silence. Either because you're afraid of going against your colleagues' gain or because you're hopeful that you will be one of the winners in the draw for the $500 million. Whatever the case might be, this company, BP, gained incredible access to the deepest and most intimate capacities that you think you still have unencumbered within your public, what you still think is your public university. Many people worry about these relationships because professors become entrepreneurs. And Jay Kisling, Jim Thomas, forgot to mention among the many, many different titles that Jay Kisling has, that he also is supposed to be a professor in a public university. He's part of the faculty in our campus. These professors become entrepreneurs, students become employees, patent holders, and so on. Many people worry about these things, but what I worry mostly is about the takeover and the privatization of the very soul and the very purpose, the public purpose, of the last line of defense that the public has to confront technological craziness. Because synthetic biology is hardly a technology, as I said, right? And if you compromise that, if you alter it in such a way that it becomes internally transformed so that it cannot do the job it was supposed to do and yet continues to present itself to the world as if it was doing that job, you really have compromised all your capacity to deal with this type of development as a couple. Many of you, and I'm going to stop because I know that Andy is probably keeping track of this, many of you might have forgotten about a development that happened in 2010, and that was a big, big explosion in the Gulf of Mexico, remember? It's been forgotten, and the reason why it's been forgotten by many is because it was declared solved. The problem was declared solved on August 24th of 2010 through a paper in Science that was published by a very large number of scientists with very good reputations and very good credentials, all of them at Berkeley. The paper in Science is a little bit cryptic, and you can say, oh well, it's not about really cleaning up the Gulf, but immediately on the same day, the New York Times was also publishing an article that starts, the first line says something like, the Gulf oil plume is no more. Problem gone. The paper is about how miraculously out of nowhere or somewhere, microbes appeared in the Gulf of Mexico, who ate all the oil that was floating around and cleaned it up, and then also equally miraculously went away and went home, and I don't know where they lived. Many of you might have heard this story. This story then comes through the most certified scientific channels, through the most public and peer-reviewed and so on channels. The New York Times validates it and so on. What you never read as a public is that every single member of that publication team was compromised by a deal with BP itself. So the very role that we should have been playing has been compromised. Our very institution has been genetically engineered from the inside. And to me, that is really the thing that I think we should all be very aware of. It has reached a point where science has become exactly the opposite of what you would expect. There used to be a time when science was the breaker of myths. And today, especially in this field, where people talk as though they could really so-called create life by so-called using so-called code and programming as if living things were computers and so on. Science, or what is called science these days, this compromised genetically engineered science, is the proponent of myths and the perpetuator of myths. Exactly the opposite of what you would expect. A most perverse result that comes from something that has happened while you guys were not watching. So it's really great to see that you're aware of it, that you're looking at it, and really I look forward to see what you will do about it. Thank you.
Thank you, Ignacio. I told you he was the conscience. Tim was saying before about the 1970s and 80s when the first wave of genetic engineering hit, and probably for me the most impressive group that began to address those concerns was the Council for Responsible Genetics. Great thinkers like Richard Lewontin and Ruth Hubbard, who's written a book called Exploding the Gene Myth, very much on the lines of what Ignacio was saying, came to fore and really became really the brain trust for those who are trying to better understand and then regulate that technology. I should also add that the CS Fund, which is generously funding so we can have this forum tonight, was right there then as well, and they started something called the Biotechnology Working Group in order to get NGOs involved. I was part of that. But the torch has passed at the Council for Responsible Genetics to Jeremy Gruber, our next speaker. And Jeremy has just done a fantastic job. If you don't subscribe to Gene Watch, which is the extraordinary magazine that the Council for Responsible Genetics puts out, please put that on your to-do list. And also, the books are coming back. I know that 2 books are being published, and I think I heard today from Jeremy that yet a 3rd book is coming out. So we know the universities and maybe even the schools of the universities are at risk. Jeremy's going to tell us what else is at risk, potentially, with this technology. Or
Thank you, Andy. So, I'm not going to be talking too much actually about synthetic biology. I'm going to be talking about laboratory safety because, of course, the reason we're all here is to talk about a new lab that's been proposed for Richmond. So my task today, or this evening, is to give you a very quick primer of what's involved in lab safety. Obviously, there's a lot involved, and so these slides will be available hopefully on the website of the event, and I appreciate the organizers for inviting me to come. There are a lot of guidelines out there relevant to different forms of lab safety. The problem is that the guidelines are very much just that, guidelines. There is a lack of oversight, a lack of enforcement, and very much a limitation in what the guidelines on different areas of biolab safety are able to do, which means that for the vast majority of labs operating in this country, the safety regulations are largely through self-regulation, not through oversight and enforcement of other agencies. So when you hear a lab, any lab, tell you that they are following the regulations, take that with a grain of salt. The regulations are limited and the enforcement is poor. So why is biosafety important? Well, biosafety is important because if the researchers in the biolabs are doing what they are supposed to be doing, accidents can happen. Biolabs are, by their very definition, a man-made venture, and people make mistakes. We have biosafety guidelines to ensure that those mistakes are minimized and that the accidents do not breach the facilities themselves. Unfortunately, they do. The goal, of course, is to ensure that we do not have workplace injuries, that we do not have property loss and unintended environmental releases and damages, but they do occur. So what is biosafety in terms of the different levels? Biosafety, when we talk about biosafety, we're talking about the way that we both construct labs and design equipment within labs to ensure that both the workers on the front lines in biolabs and the community at large are safe. The guidelines that are promulgated by the NIH, National Institutes of Health, and CDC outline 4 stages of biosafety, starting from a minimal level, biosafety level 1, labs that are working with agents that are not infectious to humans, all the way to biosafety level 4, highly infectious exotic agents for which there is no treatment. Again, barriers that biolabs must construct are both primary barriers, that are barriers in terms of what the workers themselves must have, clothing, breathing apparatuses, and then, of course, secondary barriers, actual structural barriers within the facility itself to ensure that it's safe. And that's an important distinction because secondary barriers must be considered in the construction of the facility. So when we're talking about a new biolab in a community, the biosafety level designation will necessarily impact how the lab is constructed. Again, biosafety levels, an increasing risk of health and safety to humans. Laboratory locations are, as you can see, all over. We have biolabs everywhere, both low-level labs as well as high-level labs. And so we have to ensure, of course, that these labs are safe, particularly because biolabs in the United States have proliferated rapidly just over the last 10 years. In the last 10 years, we've seen a tripling of biolabs operating in the United States. In the last 25 years, we've seen 1,500 symptom-causing infections from biolabs, resulting in 36 deaths in the United States. And those numbers, we have very poor accounting for numbers in this country when it comes to biolab accidents. The Presidential Commission that released those numbers determined that they were wildly underestimated and probably double or triple those numbers. A case in point, right here in Berkeley, workers who handled Rocky Mountain spotted fever, which is deadly, which can spread through the air, happened under improper conditions simply because the agent was mislabeled. So accidents can happen in all kinds of ways in biolabs, and they will. The lack of oversight in biolabs leads to a lack of policing, and I just want to give you 2 quick case examples. At Texas A&M University, Brucellus virus, several workers were infected with it. The university did not report it, and until local activists actually forced the district attorney to create an investigation. And the CDC discovered that on investigation, missing pathogens, additional infections, and in fact found that the CDC had actually investigated the facility earlier and found no problem. So even when there is oversight by sophisticated agencies like the CDC, it is oftentimes ineffective. Boston University, another example, 3 BU researchers were infected with tularemia. The Soviets had actually worked with tularemia a number of years ago as a possible bioweapons agent. That's exactly how deadly it is. Again, Boston University did not properly report it to the CDC. They waited several months to report it to the Department of Public Health, and the Department of Public Health found that it was a major concern for the community, that it had not been reported. And it's important also to understand exactly when regulatory apparatuses do work, how effective they are. In the case of Boston University, the Occupational Safety and Health Administration actually did come in after the fact and investigate, and based upon what they found, fined the university $8,000. So three individuals infected with a deadly virus under improper conditions, not reported to the community or public health officials, and for all of that effort, all they received was an $8,000 fine. So when it comes to synthetic biology, and this will be the one time I will talk about synthetic biology, it's particularly important to note how synthetic biology and biosafety today in the U.S. Are a bit at odds. Because biosafety levels, the way they're designed today, are based upon predictability. The predictable properties of whatever agents are being worked at in the lab, that determines how the lab is classified in terms of safety levels. But with synthetic biology, what you're doing is you're taking relatively innocuous low-level agents and you are working with them to create new properties. So the regulations are completely incompatible with synthetic biology because they look at the agents as they enter the laboratory, not how they change within the laboratory. And the emergent properties that can be created and the risks from those emergent properties are oftentimes not contemplated by current biosafety regulations. It's also a particular concern because synthetic biology, as compared to a lot of other areas of biology research, involves the input of people from a lot of different fields. It's very much a cross-disciplinary pursuit, and so you have people who are engineers and from other fields coming in. These are people without biosafety training. These are people who are unfamiliar with biological laboratory health and safety issues. And so not only do you have the science incompatible with the biosafety regulations, but you oftentimes can have the personnel incompatible as well. Environmental reviews. One of the first things that happens when you construct a biolab is you're going to have an environmental review. Sometimes they are at state level, sometimes also at the federal level. Environmental reviews are conducted initially by whatever entity is constructing the lab. In this case, it'll be the LBNL or an agency on behalf of the LBNL. Environmental reviews are notoriously limited. The state agencies that oftentimes have to review them have none of the background and experience to actually understand and appreciate the issues that are oftentimes raised in environmental reviews. And it's oftentimes only through litigation, after environmental reviews have been offered to the community, that those reviews have been properly assessed and improved upon. 2 examples again, in the case of the facility at Fort Detrick, the initial environmental review done by the lab there found no problems, no health and safety problems in the community. a review by the National Academy of Sciences found it completely insufficient, that they had not contemplated many of the health and safety issues involved in the laboratory and required that the lab redo their environmental impact assessment. Secondly, in the case, again, of Boston University, the National Research Council found that the University of Boston, in their own environmental review, had been insufficient. This is, of course, after individuals from within the community had sued, both in state and federal court, the lab based upon their environmental review. That lab then produced a secondary supplementary review, which was then reviewed by the National Research Council and found defective. So the lab did 2 reviews after 3 separate litigations. And after all that, the review, and in this case, the review is done by the National Institutes of Health, perhaps the agency with the most sense and ability about these issues. And it was still found deficient, so deficient that for the first time ever, the National Institutes of Health had to actually redo their policies in terms of environmental impact assessments with labs, because their current policy making was found to be ineffective in the case of Boston University. So these are some of the questions that any community for which a biolab is going to be constructed are going to have to contemplate. What safety levels will that campus house? What pathogens are being used for research? In the case of the lab in the LBNL lab, there are going to be different entities working, so with different guidelines. So what is the overarching guidelines going to be? What remedies are going to be available? What is the safety infrastructure with the community? If there's an accident in the facility, how is the community going to respond? Very basic safety services, fire, emergency services. How are they going to work? Is there a plan in place? Will the LBNL commit to a transparent and comprehensive risk assessment? Oftentimes, low-level labs are not required to complete comprehensive risk assessments. So it's incumbent upon the community to hold the lab accountable to do exactly that. Will the lab be developing specific health and safety procedures for synthetic biology? When any lab says that they are working consistent with current regulations when it comes to synthetic biology, that's meaningless. There are no regulations when it comes to synthetic biology. If the Lawrence Berkeley Lab wants to be on the cutting edge of science, that's great. They should also be on the cutting edge of safety and health regulations for the community. To make sure that the lab's work on an ongoing basis was transparent, because it's important to note that whatever the lab says they're going to do and whatever the lab starts doing on the first day of operation, if they begin operating, it will be very different the next year and the year after. Research changes, research develops in laboratories. So it's important that the community not only have transparency at the beginning of this process, but also that they have transparency on an ongoing basis as well. So finally, I'd just like to say that in terms of how I at least think you should be viewing this issue, this is really about what is a good neighbor. The LBNL is not constructing a housing project in the community. They're not constructing a shopping center in the community. They're constructing something very specific, a biolab involved in synthetic biology research. There are very specific things involved in that, particularly from a health and safety standpoint. The one thing, we haven't heard a lot from the lab, so we really don't know a lot. But what we do know is what they haven't said. And that's been a year of promoting the lab, talking about the lab to a lot of different communities, and a year of where they've been completely silent on health and safety issues. I think it's incumbent upon the lab to continue a dialogue with the community where they start talking about health and safety issues, because they are a real and important concern for Richmond and for any community that has a biolab and a synthetic biology lab in particular.